Hi, everyone, and welcome to today's NHLBI Small Biz Hangout, titled How Pharma Evaluates New Therapeutic Opportunities. Our presenter will be Dr. Craig Wegner of AstraZeneca Pharmaceuticals, and I'm Gary Robinson, a Business Development Advisor at the National Heart, Lung, and Blood Institute. So today's presenter is Dr. Craig Wegner. He is head of the Boston Emerging Innovations Unit within AstraZeneca's Innovative Medicines and Early Development Biotech Unit. Craig is a passionate translational scientist and strategic leader with more than 25 years of pharmaceutical research experience at 5 major pharma companies. And he's published peer-reviewed articles, edited books, and led initiatives and teams spanning a range of disease areas and disciplines. Relevant to today's webinar, Craig has headed initiatives at both Pfizer and AstraZeneca related to analyzing and avoiding pharmaceutical project attrition. As an executive director of translational science at AstraZeneca, Craig directs the company's drug repositioning ideation team. He also manages AZ's partnership with the NIH National Center for Advancing Translational Sciences, aka NCATS, and Taiwan's National Research Program for Biopharmaceuticals. He also heads AstraZeneca's expanding open innovation platform, which partners internal AstraZeneca assets and know-how with the disease insight and expertise of academic physician and basic scientists to collaboratively advance pioneering research and therapies for the benefit of patients. Craig received his PhD in biomedical engineering from the University of California, Davis, and over the course of his accomplished career, he's made critical contributions to 5 marketed drugs. It's a great pleasure to have Craig speak today on how pharmaceutical companies evaluate new therapeutic opportunities. And as I pass the baton over to Craig, I will just point out everybody should be muted, but in any event, please mute your microphones, and we will be taking questions through the Q&A panel.
Okay, thanks, Gary, for the introduction. This is a topic that I am passionate about. As Gary said, I've been involved with looking at attrition analysis while I was at Pfizer, and again here while I was at AstraZeneca. And consequently, as we and other pharma look at, and that work has been published, so as we and other pharma look at new therapeutic opportunities, we keep a lot of these concepts in the back of our mind, and consequently it's good for anyone who's on the other side trying to find a partner in a pharmaceutical company to be aware of those so you know what your audience will be looking for. So by way of introduction, despite we all have great ideas, but it's been shown that 99% of those innovative ideas will ultimately fail. Now, those that are on the call today, I'm sure your ideas are better than most others, so it may be still 70% or maybe 80%, but it's still the vast majority of those ideas are going to fail. What's been discouraging as we in the pharmaceutical companies have went back and looked at projects that fail versus projects that succeed, many times, and in fact in about 50% of those cases, we could have predicted failure prior to doing the next expensive experiment. So what that has caused us to do is to recognize those and those common causes of attrition, and now use those as we evaluate projects and try to become unbiased to those. And as I'll share today, I think it's important that you, as you're viewing your projects and ultimately preparing them for presentation to either investors or to companies that you would want to partner with, have attention to these. They will result in both the success of the project, they'll result in the timely tractability or progression of the project, and they are going to relate to the attractiveness of the project to investigators or to investors. So the real driver of looking back at attrition is shown on this slide. Relative to a successful period in the 1990s, shown on the left-hand side of the slide, there was an extreme drop in the number of approved compounds as we entered into the mid-2000s. And this was despite an increase in the expenditure within pharmaceutical company R&Ds. So this level of productivity, a decrease in number of approved drugs, at the time you're increasing your expense in R&D, obviously is unsustainable. And when we went back and looked at, well, what phase would compounds attrit at, you can see that there was an increase in attrition at just about all the stages, but the ones with the most predominant slopes, were during phase 2 and phase 3, and these are expensive times in drug development. So at the times we were spending the most money, we were also getting the highest attrition. So there were separate companies that went back and looked at this attrition, and I'll present two of those today that are published. The first was a team that I actually led at Pfizer. We looked at 44 programs that reached phase 2 and a decision point in phase 2 in the period between 2005 and 2009. And what we found that was, disappointingly, in 43% of the cases, we were not sure that the mechanism was actually tested. Now, it's one thing if your hypothesis is wrong, but it's another thing if you have delivered a compound into phase 2 and you don't even know whether you've tested that particular mechanism that the compound hits. And this led us to, developing some pharmacokinetic and pharmacodynamic, or PK and PD, principles to provide a solution to this. And I'll go into more details and give you the reference to that work as well in subsequent slides. This was followed up by a study within AstraZeneca that occurred almost at the same time frame, a little bit later than the Pfizer work, and encompassed all stages of drug discovery, where they looked at 142 drugs that reached the decision of either going forward or halting between the 2005 and 2010 time period. And the reason that they did this analysis was, at that period of time, the success within AstraZeneca was about 1/3 of the industry median. And the result of their analysis was that in 75% of the time, they found that the project had a really big risk. And so if they had eliminated those 75% of the projects and focused their investment on the 25 that didn't have the big risk, their success rate actually would have been a little bit above the industry median, as opposed to 70% below. And this led them to what they call the 5 R's, or the right target, the right tissue, the right safety, the right patient, and the right commercial. And again, I'll go into more details on each of those in subsequent slides. And finally, just to let you know what's coming, at the end of the presentation today, I'll sort of take a accumulation or a constellation of those two attrition analysis to come up with what I think are the building blocks that you should be concerned of as you prepare a project from early stage and to a stage where you're looking for a partner, either as an investor or as a company to license the opportunity to. So first of all, the Pfizer analysis resulted in what they call the 3 pillars of survival. And basically it was that in order to know that you've tested your mechanism in your clinical study, in the phase 2 clinical study, you needed evidence in the tissue of interest of, first, that you had exposure of the compound to that tissue, that it bound to your molecular target that you were trying to get it to bind to, and that finally that it produced a pharmacodynamic effect. And if you had these 3 pillars, then you had twice the likelihood of getting a positive signal in your phase 2 study. And more details on that, they went back of the 43 programs that they analyzed, or that we analyzed actually, we aligned them into this 2x2 matrix where we had confidence in exposure or confidence in pharmacology. So 12 of the programs actually ended up where we had low confidence that we were achieving necessary exposure for efficacy, and we had low confidence, meaning no evidence that we were actually hitting the target from a pharmacology biomarker. In all 12 of those cases, the compound ultimately failed, and we failed to test the mechanism. Up here in the upper left-hand corner of this matrix, where we had confidence in exposure and binding to target, of the 12 projects that got here, 5 of them we were pretty confident that we tested the mechanism, and 2 of those were successes in the phase 2 that they resulted in phase 3 starts. On the other end, in the lower right, where we had confidence in the pharmacologic effect of the 6 projects here, we were confident that 5 of them we had actually tested the mechanism, although none of them resulted in phase 3 starts. So we tested the mechanism, but our original hypothesis was probably wrong. And in the case in the upper right, which is where we wanted to be, if you had all 3 pillars, then of the 14 projects that ended up here, all 14 we were sure that we tested the mechanism. 12 of those achieved proof of concept in phase 2, and 8 of those advanced into phase 3. So if we just look at confidence in exposure, meaning that you know that you had enough compound that reached the target tissue, and in 73% of the cases, you were confident that you actually tested the mechanism. You tested your hypothesis. And in almost 40% of the cases, those projects advanced into phase 3. So your hypothesis was right in 40% of the cases, and you could advance it into phase 3. Oops. On the other hand, if you had confidence in pharmacology, that meaning you had a biomarker of a pharmacologic effect of that pathway within that mechanism, now you were almost assured, 95% confident that you had tested the mechanism, and 40% of the time, again, you advanced into phase 3. Now, just following this, and this work was all published. Boy, did I forget to give you the publication in Drug Discovery Today in 2012. So just after that, AstraZeneca did an analysis of their drug pipeline, and again, unlike Pfizer, which had focused solely on phase 2 decisions, they focused on decisions across the whole discovery pipeline, from early discovery to those in development, and looked at 142 projects. That's one of the benefits of being in a big pharma, is not only you have a few successes, but you have a lot of failures that you can learn from. And again, AstraZeneca did this because their success rate was about 1/3 of what peers were. They published this work in Nature Reviews of Drug Discovery. And what they found from looking at it was that there were really 5 key frameworks that were important to have knowledge about to advance a project forward. The first was, is it the right target? Is there a strong link between the target and the disease, particularly in humans? Is there evidence that it's going to be differentiated? Is there evidence that there's available predictive biomarkers? And I'll show you some of that evidence for why this is important as we go forward in this presentation. The second thing that they said was the same thing as Pfizer. It's important that you know that you got the right concentration to the tissue. So did you have adequate bioavailability and tissue exposure of the compound? Do you have some pharmacodynamic biomarker that gives you evidence that you've not only got the compound to the target, but it's produced a pharmacologic effect, and hopefully a maximal pharmacologic effect hitting that target? Do you understand the PK-PD relationship from preclinical models to be able to translate that into the clinic? Do you understand drug-drug interactions so that you know that what concomitant medicines to avoid or what the effect of they would be? AstraZeneca also felt it important that you needed to really understand the safety because a number of their compounds, they were limited on the exposure that they could go into in the clinic because of unanticipated safety findings. So based on preclinical data, was there a clear knowledge of the differentiation and margins for safety? Was there an understanding of the secondary pharmacological risk, i.e. the off-target pharmacological risk? And then any understanding of metabolites or genotox or drug-drug interactions, again, with the compound, as well as any on-target safety issues that needed to be dealt with. Next, it was important not only that you knew that you had all these covered, but that you went into the right patient population. And the right patient population means not only the right indication, but within the indication, the right patient population within that indication. And I'll show you some examples later in the slide deck of where we, AstraZeneca, got that wrong and where we could have done that better. And then finally, that we, at the end of the day, we're going to be able to create a return on investment. In other words, we're going to be able to recoup our development costs because of the commercial value of the agent, that it was going to be differentiated relative to the things that were already on the market or in the pipeline.
Hey, Craig, I just want to break in here and just remind people to, if you have any questions, please post them on the bottom right-hand side of the Q&A panel of the WebEx screen. Thank you. Great.
Thanks, Gary. So here's a case study of how to put the 5 R's into place. And I use this one even though it really is outside of the therapeutic area of NHLBI, but I do so because I think it gives some very good principles that then can be applied across. Other case studies that will come later in the slide deck are actually within the NHLBI disease areas. So this was a compound that was a positive allosteric modulator of mGluR2 for the treatment of schizophrenia. And the hypothesis here is that the failure or decrease in cortical glutamate drive results in a decrease in subcortical dopamine function and consequently psychosis. And at the time this project was brought forward into clinical studies, there was a Lilly compound that was a dual mGluR2 and 3 agonist that had some positive effects in a phase 2A study. Now that later didn't pan out in phase 2B or phase 3, but it led to AstraZeneca rushing this compound forward. Now, what do we know about this compound? Well, first of all, as I mentioned, it's a mGluR positive allosteric modulator, which means that it functionally enhances the effect of glutamate on the mGluR, glutamate receptor 2. It was active in 7 different preclinical antipsychotic activity models, as well as 2 anxiety models. the effective concentration in those preclinical models ranged from 2 nanomolar to 1,000 nanomolars. It did have some preclinical toxicity. It caused testicular tox in dogs and some cataracts in rats, and this capped the allowable exposure in the clinic. The phase 2 was based, the exposure target to reach in the phase 2A study was based on a collection of what we found in the preclinical model, so that somewhere between the 2 and 1,000 nanomolar. We, I think, picked closer to the 2 than we did the 1,000. And then we knew that there was that drug actually got into the central nervous system, so we knew that there was some exposure to the target tissue that was 50% of the plasma, which is pretty good, but we had no PET ligand or any other way of assessing that we'd actually hit the target in the CNS. The outcome of the phase 2A study in schizophrenia was that it was negative. So now if we look at the 5R framework, we can say, well, first of all, there's a huge market for schizophrenia, and the existing drugs are not very good. So it's a huge commercial opportunity. So this one we would say is green. But when we looked at some of the others, well, is this the right target for schizophrenia? Well, first of all, the Lilly compound had been a dual agonist of 2 and 3. We were going forward with a 2. Was that going to be effective? And ultimately, there wasn't a lot of evidence that that was going to be effective. So in retrospect, we would have graded right target as red. The 7 preclinical models, again, showed a wide range of efficacy, and the translation of these models is known to be not very good. So the other thing is that schizophrenia is a very heterogeneous indication, and we went into all comers in the phase 2A study. So did we have the right patient was the other concern that we had. In addition, what about tissue exposure? Well, we knew it got into the brain, but we didn't know how much we really needed. Some animal models, we had efficacy at 2 nanomolars. Others, it took 1,000 nanomolar. So what's the right concentration here? We were limited by the testicular tox in dogs, so we could only test up to about 15 to 20 nanomolar in the clinic. Would we have gotten efficacy if we went higher? And then the right safety, the same thing, because we were limited by the testicular toxicity. Did we need another compound? Do we need a better compound? AstraZeneca actually went back and got a better compound and is bringing that into clinical trials now for other indications. So let's talk about a few other case examples that are more relevant to the therapeutic area that those on this call are probably most interested in. So the first program is that of 11-beta HSD1 inhibitor. 11-beta HSD1 prevents the conversion of cortisone to cortisol. There's some preclinical evidence that this would be effective in diabetes and obesity. However, when we went into the clinic, we found that the activity was quite weak. This was a situation where I'm not sure we didn't have the right target. We didn't have evidence that it was differentiated or that it had the efficacy horsepower to produce efficacy that was going to be clinically meaningful in either diabetes or obesity. The second molecule that I mentioned is the glucokinase activator. Glucokinase activation catalyzes the conversion of glucose to glucose 6-phosphate. So when you administer this to animals and to humans, you get a strong acute decrease in plasma glucose, so exactly what you want in a diabetic patient. But this effect is not sustained. Again, going back and looking at this in preclinical models, had we done models that were more than 2 weeks in duration, we would have found that this effect was not sustained. So again, we didn't have the right target here. And then finally, in the lung field, we took a neutrophil elastase inhibitor into bronchiectasis, CF, and COPD. We set the dose based on some preclinical models where we exogenously added neutrophil elastase topically, intratrachely into the lung, and peripheral blood inhibition of neutrophil elastase activity. And what we found was that we had some weak efficacy in bronchiolitis, but when we went into phase 2B studies in CF and COPD, which were our primary indications, there was no effect. And we really here are unsure whether we have the right exposure within the tissue. Now, each of these also could have been a problem, did we have the right patient? And I mention that because one of the things that my group is involved in is drug repositioning, and each of these compounds are now being pursued in another patient population where we think that they have the opportunity to succeed, correcting for the right target, right indication, and tissue exposure issues that occurred in these studies. So the important thing for each of you to understand is actually the consequence that this analysis has led to, not only within AstraZeneca, but throughout the pharma industry. And that is that the concept now, if you look at the pharmaceutical drug funnel from idea to launch, you know that we, in the past, and particularly in the late 90s and through the first decade of this century, we spent a lot of time just entering as many good targets as we could into the pipeline to hope that additional compounds would come out the end. What we've found now is that we can't afford to do that, and that what we really need to do is to focus on those projects that have the most green of the 5 R's. And to focus our efforts on the other projects of either moving them into this area or not doing them at all. So the result is that if your project doesn't sit up here with most of those 5 criteria that I've mentioned being green, you're not going to get the attention of big pharma going forward. So let me just reinforce some of this with a few more case examples and sort of run through how I now think of attrition and how to build a successful project. And I build it with building blocks as shown here, starting with confidence that you've got the right compound and the right molecule against your target, that you're administering it at the right dose to cover target and to have acceptable safety, that you have the right patient population in terms of the indication that you're going after, but even more than that, that then you build on that and look within that indication into the right subpopulation of patients within that indication that are most likely to respond to your compound, respond to inhibition or activation of that particular mechanism, that you've done your clinical design to not only prove your hypothesis, but gain as much information about your drug and that indication, that patient population as possible, that you have a clear regulatory path and have talked to regulators on how to move this through to market. And this is particularly important if you're in novel therapeutic areas, which is where the highest unmet medical need and hence highest value is. And then finally, you understand the return on investment. In other words, that if someone invests in this molecule, at the end of the day, they'll be able to recoup their investment costs. So let's quickly walk through each of these. There's a lot of information on each of these slides, and I'm just going to touch on a little bit of it so that we leave some time for Q&A. But as Gary mentioned, these slides are available to you to peruse after the webinar, and if you have any questions, don't hesitate to reach out to me. So first and fundamental is you got to have the right compound or the right molecule. This is the foundation. You got to get this right. The first feature is you got to have the right potency. If you're talking about a small molecular weight compound, it needs to be in the nanomolar range, not in the micromolar range. If you're talking about a monoclonal antibody or a biologic, you need to be in the picomolar range, not the nanomolar range. Just some examples of this: anti-IL-18 antibody, not only by AstraZeneca, but by Pfizer, Abbott. I think there were 3 or 4 other companies that created antibodies to IL-18. None of these were able to be even brought successfully into phase 2 studies because they lacked the potency. They were in the nanomolar range, and they didn't cover targets sufficiently in phase 1 to go forward. Anti-C5a antibody, great target for acute lung injury. Unfortunately, there's so much C5a that you can't practically administer enough antibody to cover this target. You have to know that you've got the right selectivity and the right safety profile. Selectivity against other targets of similarity to make sure that you avoid the known adverse effects, but also that you've run a panel of receptor and enzymes and ion channels to make sure that you have no unknown safety issues. PK/PD, probably the most valid use of animal models. I'll be critical of them in terms of picking the right target, but in terms of picking the right exposure, being able to confirm that you can cover the target in animals and that produces a pharmacodynamic effect, ideally by a biomarker, but also could be by an efficacy signal, a symptomatic signal, allows you to set dose for going forward, confirms your route of administration, confirms your tissue goal. And I often get this, particularly for respiratory drugs, well, if it's not well absorbed or if we've got some systemic tox, let's just give it by inhalation. Well, this isn't an easy solution. First of all, if you're going to do a compound by inhalation, you almost have to design it to be a compound by inhalation. All of you that are in the lung field were taught early on in your lung physiology that the surface area of the lung is about the size of a tennis court. Well, now you're going to aerosolize a compound onto that tennis court that's got a thin fluid layer over it. And that compound is going to have to disperse over that thin fluid layer. It's got to be sub-nanomolar potency if it's going to hit your target. Also, the lung, things go right through it. So if you're going to keep it on that target, it's either got to be non-competitively binding or slow off rate of that molecular target. And then finally, if you're going to avoid systemic toxicity, you would want it to be rapidly metabolized or or eliminated. And then finally, it has to be formulatable in a device that you're going to administer it, use to administer it, either as a dry powder inhalation or as a meter dose inhaler. So a drug optimized for oral delivery that fails because it's not very good in terms of oral absorption or has systemic side effects, the solution isn't immediately, oh, we'll just give it by inhalation. And then finally, you have to be aware that other people are often working in the same area. So how do you stack up relative to your competition? If you're selling this to an investor or to another company, they want to know that you're the best. You're better than the competition. And so you need to give them some concrete reasons why at least you're as good as anybody else out there, if not being the best. And that means that you've also got intellectual property to cover that agent.
Hey, I just want to jump in, Craig, and just remind people that they can submit questions through the Q&A box at the bottom of the WebEx screen. Thank you.
Okay, so now that you've got a very good molecule, the next step is you have to decide on its dose. And as I mentioned in the previous slide, you need to have really strong preclinical data around its PK/PD. And a great story around this was the leukotriene story in asthma. A company by the name of Revlon, which no longer exists, was the first to get a compound to this very hot target in the late 1980s, early 1990s into the clinic. And they had a pretty good compound. They brought it into phase 2. It shifted the inhaled LTC4 dose response by 3x, okay, threefold higher concentration of LTC4 to produce a bronchoconstriction, a decrease in FEV1. They went into asthma studies and failed. At least half of the pharmaceutical industry dropped their leukotriene programs based on this Revlon finding. In addition to Revlon, they dropped and said, you know, we've tested the mechanism. It's not active in asthma. We don't know why, but it isn't active. At the same, a few years later, actually a year to 2 years later, behind them was Merck, who actually took the Revlon lead compound and made it a little bit more potent and slower off rate. And the most important thing is that they had preclinical PK/PD to know that a threefold shift isn't going to do anything in allergen-induced models preclinically, but a 30-fold shift is. So when they went into the clinic, they targeted this 30-fold, selected a dose that would produce this 30-fold change in the concentration of LTC4 to produce a bronchoconstriction, and they were effective in asthma. The compound we know as Montelukast or Singulair is a $3 billion product right now for Merck. So it is critically important that you select the dose right and that you get it right, and preclinical PK/PD can help you do that. Translational biomarkers even better because now you know that you've covered the target in humans, and of course you need the safety biomarkers are helpful as well. So this is the point where I'm going to start moving a little bit quicker and just capture a few keys on some of the slides, because if you get these two right, the rest of the things sort of fall in place. You need to pick the right indication. Human pharmacology, human genetics, human pathway biomarkers, and omics are much, much more valuable than a validated animal model. And that's why the ... to 99. Here's some data from the AstraZeneca paper that shows that if you had human genetics, that 73% of the time your project advanced to the next stage, was successful, still going on. If you didn't have human genetics, only 43% of it, given everything else being similar. If you had pharmacodynamic biomarker, it was even better. 82% of the time it would advance versus 29%. Selecting the right patient. There's lots of techniques now to know that not all asthmatics, not all patients with congestive heart failure, not all diabetics are the same. And in fact, this, with some work that was done in Joel Dudley's lab, he's now identified 3 distinct populations of diabetics. And again, the AstraZeneca work, if you were able to stratify the patients within an indication, 90% of the time you were successful. If you went into all comers, 22% of the time were you successful. Here's another example, 5 Rs. TH2 antagonist for asthma. TH2 antagonist is a receptor for PGD2 released from mast cells on activated T lymphocytes and eosinophils. AstraZeneca went into a clinical study in phase 2A study in moderate to mild atopic asthmatics. Had very, very positive results, completely inhibited the morning peak expiratory flow after withdrawal of inhaled corticosteroids, and this and also a 75% reduction in the decrease in FEV1 after the withdrawal of inhaled steroids that these patients were on. Their next study, however, they switched and went to moderate to severe asthmatics and added treatment after a run-in period. So these are now on top of inhaled corticosteroids. The result was that they only had mild efficacy, but in post-hoc analysis found that it was efficacious in atopic patients as opposed to non-atopic patients. So their phase 2B, and it was a massive phase 2B, 6-month study, now switched patient populations even more and went to moderate to severe, now atopic patients, based on the phase 2A results, but these were patients that had ICS and LABAs on board. Now, since one of the main things that corticosteroids do is decrease eosinophils and decrease lymphocytes, and this mechanism is supposed to exactly do that. If you add the compound on top of those, it's just not the right patient population. Why was this done? Because AstraZeneca wanted to go for what the medical need was and the commercial need was, so they had the wrong commercial. And if they wanted to go into that patient population, then they had the wrong target. So you need to get, back to the previous slide, you need to get the right patient population to do the study in. Quickly then, the right design is critical as well, learning as much as you can from the clinical study, the regulatory path that you're able to map out how you're going to, the line of sight from where you are now to registration is important. And finally, that at the end of the day, you're going to be able to recoup the development costs, that there's a positive return on investment. In summary, the attrition analysis that has been done has led to this enhanced rigor in selection of the programs that pharmaceutical companies are going to invest in. And so each of us need to realize that as we're bringing forward projects to venture capital investors or to pharmaceutical companies. If our projects are not in this green area and have mostly green, then we need to figure out how to move them into that green area or select another target that is there. The result of this analysis is that there's been a reversal in the number of drugs that were approved. I remember I showed you at the beginning the decrease in approval rate and the increase in costs. Well, costs now within the pharmaceutical company have leveled out because budgets just won't, income just won't allow the R&D costs to increase anymore. But you can see that the approval rate has started to increase. So if you'll follow these confidence building blocks, you're going to create a swell of enthusiasm, a volcano of heat and smoke around your projects that's going to lead to a lot of investors and a lot of other collaborators and pharmaceutical companies being interested in your project. And I thank you for your attention and open it up to a Q&A.
Thanks, thanks, Craig. That was great. Most of the questions were, how do I get a copy of the slides? Because a number of people, I guess, joined a bit late. So I'll remind everyone that when the webinar is completed, there will be a feedback form that will pop up on a separate browser window. If you complete that form and enter your email address, I'll be happy to send you a copy of the slides. So with that out of the way, let me jump into some of the questions that we've gotten. You kind of answered this in the case of the repurposing of, I forget exactly which compound it was, but so the question was, if you pick the wrong patients from the first attempt, does it kill the drug development process? And the second question, is there a difference in the answer depending on whether the drug is from a big pharma company versus a small pharma company? So I could help with the second question, but maybe with the first one, if you pick the wrong patients the first time around, what effect does that have on the process?
It actually has a huge effect. You'd hope that you'd learn from that and would go back and get a second chance. But I can tell you there's very little patience, at least within big pharmaceutical companies, for second chances. We've got other programs that haven't had their first chance that executives would rather invest in. So you're much better off getting it right the first time. Having said that, that is why when you do the study, you want to gather as much information as is reasonably feasible, because that additional information might open up a clue to a better indication, a better subset of patients, or even a better indication to go after. And even if it isn't the company that the compound started out with, oftentimes a group of the project team will get so excited about that learning that they'll spin it out and take that additional indication forward, and that's happening more and more today. Yeah. So no is the answer, but it's a qualified no.
Yeah. So I think with small companies, I think there are a lot of, they can't always afford to run through the same sort of rigorous analysis. And so they often will make bets on compounds that may fall short in a few areas, hoping they could tweak the pharmacokinetics or the bioavailability or, through a formulation. But I think smaller companies would probably be more inclined to take risks on molecules that don't fit the right profile. Would you tend to agree with that?
I would absolutely agree with that. And in fact, there's even one large pharmaceutical company, Novartis, that has a strategy that it's very difficult to pick the right indication, let alone the right patient population in your first study. So they will do 3 to 5 small clinical studies in separate patient populations or separate indications, and then use that data to advance forward. That's unusual within the industry, but they've shown some clear success with that strategy.
Okay. Another question is, are some of the questions relating to the 5 Rs better asked at specific development stages? for example, during preclinical development versus phase 1? And should you keep moving forward if you don't know the answers to each of the 5 R's at each of the stages?
I think it's a cumulative progression on each of the 5 R's as the project progresses. But the answer is that you should have an idea about each of them from all the way through the process, from the very beginning to the very end, and continuing to test your hypotheses and to try to strengthen your R's as you go through.
Okay. There was another question on an AstraZeneca drug. Could you comment on the history of Zymelogatran, and how did the company fail to detect the liver toxicity prior to human trials? Do you know about that?
I'm not competent and able to answer that question, no, sorry.
Okay. There was a question about how pharma companies view biologic treatments for rare diseases in terms of their interest in developing them. I guess, and a related question on what size of a market will justify a return on investment. I guess it's the question of how big of an indication really does it need to be for you guys to have an interest?
So it varies on the company. Most of your big pharma, your top tier companies will require some peak year sales, certainly north of 250 million and sometimes north of 400 million. But there's niche biotech companies that are very enthusiastic about $50 to $150 million markets. Now that's total sales. Of course, it depends on the development costs, on whether that's ultimately going to be a good return on investment. But for rare diseases, usually the studies are small and not as long, and the safety packages that are required for registration with the FDA are not as large, so the costs of development are smaller as well. So it really depends on the company.
Okay.
And this is where knowing your customer becomes important.
Another question is the use of imaging studies to segment patients for treatment and following early responses acceptable to pharma in general, or I guess I'm parsing this in general, or is it undesirable because of a restriction on labeling or a potentially expensive gateway?
Yeah, that's an excellent question. Certainly for phase 2 studies, we use it all the time. It's very acceptable. We would hope to be able to convert that to some sort of diagnostic kit so that it would be universally available and inexpensive if you got the compound to market. But the most important thing is identifying the patient population where you have clear efficacy early on in your development, and usually you can solve those other problems as you go forward.
Mm-hmm. Okay.
As a diagnostic to put a patient into a once you have a marketed drug, then it becomes restrictive.
Yeah. Let's see. Question about how do the building blocks, I guess the various kind of steps that you presented apply to repurposing of existing drugs, especially if the repurposing is being done by a business or lab other than the one that originally developed the drug?
So I think they're crucial in that if you are trying to repurpose a poor compound or at the wrong dose, you're going to run into the same attrition issues that the primary project ran into. But if you have that very solid foundation, then they're critical. Then they offer you a clear advantage. And for example, there's when we entered into our relationship with the NCATs and Francis Collins felt you must have hundreds of drugs sitting on your shelf. Well, it's true, we have hundreds of drugs sitting on our shelf, but after going through phase 2 studies, we know of only about 20 of them that cover target, have a good safety index, and are consequently suitable to testing in a new indication.
So another question is, so a lot of this, what you're describing is a sort of an analysis of a drug, a post hoc analysis of drugs that you would apply in a pre-hoc approach, but it leaves open the question of how to decide on what an appropriate target is, or what ultimately a good target is, and what is your thinking, or what is the prevalent thinking about the best way of identifying good disease targets?
Human functional genomics, or human functional omics, meaning that you have human data that shows that pathway is perturbed in disease, and that ideally through genetic experiments of genetic nature or through other pharmaceutical agents, you have some evidence that blocking that pathway will ultimately lead to a positive effect. Absent that, simply that pathway is altered, either positively or negatively in the disease in humans.
Okay. And the final question, often you may get positive findings in preclinical studies, as you noted for AZD 8529. What could have been done differently to better assess the target?
Well, certainly making sure that you understood what concentration in the preclinical models is required to produce a pharmacodynamic effect that you're looking for that's relevant to humans, and then achieving that in the clinic. And the translation of animal models of disease to the human disease situation are just so poor that it's really, sometimes you just have to go forward and test your hypothesis in humans.
Okay.
But if you're going to do that, you have to make sure that you're going to test your hypothesis. In other words, you have a compound that's going to hit your mechanism, and that at the end of the day, if it doesn't work, you can walk away from that and move on to another indication or another compound, another mechanism.
Okay, thanks. So another note on the feedback forum to be able to get some slides. I want to remind everybody that the NHLBI Small Business Program has a variety of resources and opportunities we want you to know about, and you can reach out to us in a variety of ways as described here. And a final disclaimer that these are organized by the National Heart, Lung, and Blood Institute and are directed for folks who are applying for or receive funding through the NHLBI. If you're developing products outside of our mission space, you can reach out to program officers in the appropriate institute, and you can find those individuals on the central SBIR website for the NIH. Craig, I want to thank you very much for a really great presentation, very informative, and thank you to everybody for listening and attending, and hope you tune in to a future Hangout. Take care. Bye.